All right, so my name's Chris Jones. I work as a scientist here at ARI, and I'm a botanist, so my talk is going to be a bit plant biased, even though I do care about other things as well. And so my talk is focused on the nitty-gritty of the data, the monitoring part. So this is really focused on one piece of the process that everyone's been talking about today. And this fits into any process, so whether it's the open standards or you've got some other CAP or MER plan or adaptive management and structured decision making, they're all very good ways of thinking about your management problem, and monitoring slots in that as one piece of it. All right, so unfortunately, as you're all very well aware, there's no single answer for monitoring. You can't present someone with a problem. We want to monitor something and they'll say, yep, the answer is this. This is what you should do. Which is a shame because it makes it a real challenge, and this is why we're here. It's not the easiest thing to do. But there is an upside. While there are monitoring methods that you can use that are not the best, where you won't really get the data that you need and might be a bit of wasted money or effort, there are usually multiple ways of getting the right data. And so you can have a few different options and they're going to be relatively equivalent. So you don't have to stress too much about, is this exactly the right one necessarily, because there's usually multiple options to get your good outcome. All right, so I'm just going to show you a little bit of example of just some general monitoring type things in this place that we want to monitor. This is just a patch of bush with a creek running through the middle, and we can do all kinds of things in a place like this. We might be interested in the EVC mapping, and so we map out the different patches or habitat zones, one along the creek and a few different other ones, and we mapped it out with our GPS and give data within those patches. We might be doing transects, and so we put our transects throughout the area to collect data on that area. We might actually be more interested in the creek itself, and so we put our transects in relation to the creek spatially to collect data there. We might not actually care about the whole creek. We might care about a few particular spots on it and have a really specific question about distance away from the creek, and so we have a different arrangement. Maybe we're more of quadrat people and do quadrats instead of transects, and so we spread quadrats throughout the site. We use small ones because they're quicker, or maybe we have a different design, have a same sort of number of quadrats, but we want a really structured method where we can go to a point and have a really structured layout of the design. So because we're looking at a different sort of question, maybe we're interested in at different points along a transect, are we getting variation in vegetation? Is the quadrat up this end of the transect very, very different from the one at the other end, but it's more similar to the ones closer to it? There's different things you can answer with different types of data. None of them are wrong, they're all doing different things. Alternatively, we could do big quadrats. The small ones weren't appropriate because we wanted to look at bigger things, trees or something like that, and so we need to do bigger areas. There's all different ways that we can look at it. Woody weeds is one that was mentioned a bit earlier. That's a very common management action. We want to manage our woody weeds. In this case, we could go out with a GPS and put a point on every single weed that we find throughout the site, and mark them, and we can track that. We might actually care about the specific species of weed, and so we separate the data into different categories. Maybe for the willows, the yellow dots in this case, they're all clustered together. We don't really care about the number of individuals. We're really more interested in the area of willows. That's what we care about. So it's more appropriate to do a polygon type of thing. And so you're collecting polygon data in this situation and point data for your olives. And again, it's not wrong. So when it comes down to it, your monitoring design and what you're doing with your monitoring is really based on your specific context. We might be doing river surveys and we care about the fish, but we don't care about anything else that we find in there. And so we need to make sure that our data is collecting that. So there's a few questions that we need to think about. Obviously, the first one is why monitoring? This is our objective. It's our KEA, the key evaluation asset, or our KEQ, evaluation question, call it whatever the term it is you want. your objective. And then what are you monitoring? This is the target or your indicator, the thing that you actually are collecting data on. But then often we don't take these next couple of steps of how certain do we need to be? What data are we going to get? How are we going to use the data? And what resources do we have? So I'm going to go roughly through these things in the talk today. And finally, how we will use the information. So I was thinking of an analogy before about monitoring, and I think for me, what works is thinking about it as a crop. You can't just chuck some seeds on the ground and stuff comes up and you harvest it and you've got your data, you've harvested your data. And that's unlikely to work very well. But if you plan it very well, you design it well, you test if you're not sure, then you do it and then you harvest your crop, then you're much more likely to get a better yield from your data. However, we don't want to just put all our harvest into a silo and let it sit there in a filing cabinet or a shelf or a file on the computer and not do anything with it. We're not getting our value. It's this huge asset that we've got that's just sitting there and is not being used. And we need to do something with it. We want to make something with it. We want to learn from it. We want to achieve something. And that's a really important step, to not let it sit in the silo, but to do something. All right, so firstly, why are we monitoring? Our objectives. And it's important as a reminder here that it's not your management objective. So our management objective might be to remove woody weeds. The monitoring objectives is not to remove woody weeds, it's to monitor the reduction in woody weeds, or to have we gone towards a target that was specified. And so make sure that objective is specifically around the monitoring. This is not about the management itself. So a couple of examples here are to detect change in condition of something, usually vegetation condition or whatever it might be. You might be wanting to measure the increase in native vegetation cover, to verify that we've eliminated our woody weeds from a site, to record occupancy of critters in nest boxes, or the presence abundance of a threatened species. Lots of different objectives we could have for our monitoring. You don't have to have an objective for every single management objective. You might be managing for 10 different things, but we only have the resources to monitor one of them. And so this is where our monitoring objective comes in here. What is our monitoring specifically trying to achieve? So what are we monitoring? This comes back to our targets, our indicators, the thing that we're measuring. So for these two examples of objectives here, one for occupancy of nest boxes and one for elimination of woody weeds, we might have a target for the first one for the frequency of use by our particular species of interest, threatened species or something. Whereas for woody weeds, it might be the count or amount of cover of a woody weed. And there can be multiple targets for an objective as well. It's up to you, but if they're specified, you're going in the right direction. That's the key here. So it's important to make sure that your target is actually meaningful and measurable. You don't want to specify something that you can't actually measure or track very well. You might be really interested in something, but it's just infeasible to collect the data on that. Costs too much, takes too long, whatever it might be. An example here is vegetation condition. I thought I should talk about this specifically, and it's really common to have a management objective of improving vegetation condition. And so logically, the monitoring that goes alongside that is to monitor improved condition. But as everyone knows, and we keep hearing all the time, vegetation condition is a really broad term and there's lots that goes into that. And so it would be great if we had something that measured condition as a thing, and it's like, yes, we do, it's the habitat hectares or the vegetation quality assessment. It's like, yes, that does assess condition, but it's not a monitoring tool. It wasn't designed for that. It's great to give snapshots in time. You've gone to this site, you can compare, this one is worse condition to this site. They're different vegetation types, but we can see which is relatively a good or bad one. And it's doing that objective really well. You might use it in a broad monitoring sense in terms of we want it to on average, our condition scores to go up through time. We know that they're starting at, these ones are really low, we want those to get higher. But you have your answer at the end, it's got a habitat hectare score of 10 and now it's 20. That's an improvement, that's good, but we don't really know what that means. And that's the problem with using a metric like that to evaluate condition. So that can be, just be careful about using that, I guess is the idea. So in reality, vegetation condition is a composite of heaps of things that you care about. For trees, for example, it might be the cover that you care about or the density of trees. The species, the size, maybe it's just the hollows that you care about. Understorey, cover, diversity, how much, the relative proportion of weeds, or the ground layer, or what's going on there. And so it might be the case that what you actually care about is one or two or three of these things, rather than the whole condition. So if you've got on your monitoring target, we want to target vegetation condition, I encourage you to look a little bit deeper about what the things you want to monitor. And it may come back to you might come to the end of that and say, actually, we do want to monitor all of it. We want to know the condition. We want to monitor everything. I say, okay, great, that's really good. But let's devise methods that monitor all of these things appropriately. And make sure that you have your purpose of why you're trying to measure. Are you trying to monitor this condition? If it's in better condition, it's a more resilient system. If it's in better condition, it's better for fauna. Maybe your objectives are around fauna rather than the condition of the site in general. So that comes into it. So an example of this process where you can look at a whole collection of things for condition is a little thing here, the Eastern Alliance of Greenhouse Action, the EGA, group of councils put together, and they've got a bit of a plan. And they use a monitoring approach where they combine monitoring of many of these things to get it at a condition type metric, and I was involved to some extent in that one, and there's information online if you want to follow that up. Okay, great. We've got our monitoring objectives and our targets. We can just go in and monitor and collect data and that sort of thing, and obviously we can't just dive in and that sort of thing, and Rob mentioned it earlier, it's like, it should be pretty straightforward. We want to monitor, let's get a method and collect data and that sort of thing, and we'll be right. It doesn't really work like that. And so what we need to think about is, we really need to think about the data and how certain it needs to be. And this really influences the design of our monitoring approach, the methods we use, how we analyse it, and what we do with it, what our purpose is. So I'm going to go through a bunch of slides here about a bit more about the data. And so hopefully I can get you thinking about the data that's going to come out of your monitoring. So we've gone back to our spot with our creek in it, and we've got a patch that we care about in the blue at the top. We've surveyed something in this patch, and it's given us a score of 10. This could be anything. It could be a count of trees, it could be a cover of grasses or something like that. Doesn't really matter. And then we look across a few different patches within this site, and we've got 3 different scores here for different cover. So we've just, in this situation, we've gone and made an estimate for that whole thing, and we've got no idea of uncertainty. So we think that the middle one has the most, and the third one has the least of this thing. But we don't really know. Are they actually different? Are they really similar? Can we confidently say that the middle one's the highest or not? And with just this point information, it's not really very informative. It's hard to use this information to do anything because we're not that confident. So this is why we do quadrats or transects or subsamples within a site. So we've gone and put some quadrats within our sites. We've got 3 for each, and we look at the data across those 3, and there's lots of variation, and we've got error bars around our points now, which is great. So it shows us that we are really uncertain. Before, we didn't know how uncertain we were. We could have been really certain, but we didn't know. Now we've got a little bit of an idea about how much uncertainty we have. And we can see that we actually have no idea which one's the highest or lowest. It's possible that they're all the same, and it's just the way that the data has come out that they look a bit different. So that's why there's this incentive to collect, you change your sampling design. We can collect more data, reduce our our uncertainty, and now we actually know this middle one is actually the highest value, and the bottom, the third one is actually the lowest. And that's really important in this situation to know what's higher and lower. We can actually go be confident about what we're trying to do here. This is a comparison between sites, but in monitoring, often we're looking at change through time, and so we might go back to these 3 sites patches at a second time to look at the change. And if we've got a really big change, it's really easy to see at time 2 in the pink is we've had a big reduction across all those sites in this thing, and it's great. We're confident, we've got great data that says we've got a reduction here. That's all well and good, but if you get a very small change in your site, even having reasonably good data still might not be enough to tell if there's a change there. And so if you really need to know if you've got an increase or decrease in something, it's really important to know what sort of change you're expecting to see. If you're going to a site and there's no trees and you're planting trees and you want to go and monitor cover of trees down the track, you know you're going to go from 0 to something that's more than 0. It's the most easy thing in the world. You don't have to get really accurate data to look at that signal. But if you've got looking at the effect of climate change on grasses in a single year, it might be a subtle change. It could be really small things and could be small proportions. You need pretty good data to be able to look at small things. So thinking about the size of the change that you're likely to see will really influence the amount of data you will have to collect to be able to get your answer. So thinking about the data. So monitoring change over time is sort of the name of the game. That's what we're really into here. You can just monitor it at a single point, just assess across sites, but usually we're looking at a through time type of approach. And it's hard because you've got to collect data, it's expensive. Do you go every year? Do you go every 6 months? Do you do it every 10 years? How do you plan that? There's a lot of challenges associated with that. Again, so these are the things we need to be thinking about. How much change are we expecting? If there's a big change, it's a lot easier for us. But if it's a small change, we've got to really think cleverly about how we're going to detect that change. How confident do we need to be? If it really doesn't matter, if you can think, yeah, we're sort of confident that we've got an increase and that's good enough for whatever purposes you need, then great, that's good. But if for whatever reason it's really crucial that you need to have that answer and you need to be confident about it, you're going to have to collect the right sort of data to make sure that you have that confidence. Again, it's back to resources. More data is more expensive, so you can be restricted. And attributing change to management is a real key one here. You're doing something, you want to associate it with your change to that thing. You've controlled weeds at a site where there's also been a fire and something else has happened, and then you monitor and there's been a reduction in woody weeds. It's like, so was that through the fire did that, or the weed removal did that? We're not really sure. You need to be able to think about your design to make sure that the data that you're collecting will help you answer the problem. So I've got a couple of examples here about change through time monitoring. Again, at our site, we've just gone to this single patch here, and we've collected some data. We just want to monitor this something through time. And we've gone to survey 1 and survey 2, we've got an increase over time. Great, looks like it's going up. We can be pretty happy that we're on an upward trajectory, right? Maybe. Sorry, I didn't get rid of that. I don't know why that thing's there. But I'll just... Sorry. Make sure that's in the way. Great. Anyway, doesn't matter. Yeah, no, I can't get to... Oh, the one under it? Oh, that one. That one. The orange one. Great. Now I've got this thing. think we're good. Okay, so our trajectory is going up. We're pretty happy with that. But with any monitoring data, you're collecting data through time, you need to, like 2 points in time is rarely enough to tell your story. If we collected more data in this site, now my clicker's not working. Great. We find that the peak was actually behind that point and it's gone up and now it's coming down. And so maybe through time, going forwards, we're actually on a downward slope. And so just need to be careful about how you're thinking about the data. 2 points is rarely enough to make plans about your trajectory of where you're going. And so this comes into your analysis approach as well. Okay, but often when we're doing change over time, we're not just generally monitoring something we're not doing anything. Usually we're doing it in relation to a management action. We're planting trees, or we're spraying something, or we're doing whatever, we're putting up a fence. And so at some point in time, with our arrow here, we've got our management intervention. We've surveyed just before it, which was great. Someone suggested that was a good idea, and so we collected data before we did our thing, and then we collected data after, and there's some change. It's like, this is good, there's some changes happen, but this is attributing the response to the management. Do we know that it was that? Maybe something else happened. There might have been huge rainfall in that period of time. It's hard to know. And this is where our control treatment design type situation comes in. It's very hard without it. So here we've got a red patch next to our other patch where we haven't done some works. And that's our control site. And as a good control should be, it started off in a similar condition to our managed site, and it has changed a little bit, but not as much as our other one. So this gives us much more evidence to say that the thing that we did is having a response. And so this is the Backy design, before after control intervention, is that right? For assessing change. That's really good. And the more of these sets that you have, again, the more evidence you have for the data. However, what's often the case is that our control site is actually not that similar to our starting site. So in this case, it's starting at a lower point for this variable, whatever we're looking at, and it increased, but it increased a little bit. So now we're in a really tough situation. So is that smaller increase on the control site due to the fact that it started lower? Or is it, and so it actually changed the same way? So that's really tricky, and it highlights the importance of, if you're having a control, you really need to make sure about that it's starting similar, starting conditions are similar to your managed site, or that you have some other way of accounting for this challenge if it presents itself. It's hard to know there what's going on. So in some cases, we don't have a control. This is actually really common. We can't find a control site. There's either not available, or for whatever reason, we just can't get our hands on a good one. And so what can we do instead? And there are other approaches, and a common one is doing multiple assessments before and after to look at your trajectories. And so here we've only got 2 before and 2 after, but the idea is that you've got evidence to say that there's no change prior to the management action, then after the change you've got a response, and it's on a different trajectory afterwards. And obviously the more data you have before to inform that pre-condition situation, and the more data you have after, really helps that situation to give you more confidence in what you're looking at. So objective targets, our design is getting better. We've got a control, or we don't need a control, or these sorts of things. Now we can monitor, we can do it. No, we can't still, because they still need to think about a few more things. And this is why it's really important, because any of these points you can start to go astray and collect something that you don't, that you're not happy with, or is not going to collect the data what you need. So this comes down to the bit that a lot of people go straight to at the start. What are the methods? We're going to whack out quadrats or transects or count stuff. What are we going to do? And there's heaps of different things, and quadrats and transects are very common for vegetation. But what size are we going to do? and what is measured within them. And how many will we do? Alternative things are counts for plants or animals or whatever it might be, or maybe it's a GIS data thing. So there's lots of different options, lots of things we can do to collect our data. There's plenty more than what's up here. So I'm just going to go to an example here, because we can't go through all of these things, but just looking at the cover of understory plants. It's a pretty common thing, and it's a challenge for people to monitor usually. So we're going to put a quadrat out. And here's a quadrat that we've put on the ground, and we've got some plants in there. You like the pretty simple plants, but you get the point. So usually we assess cover of plants in there. We can assess the abundance of something. There's 2 of these plants. Great, that's count, that's good. Cover is much more tricky. And if I got everyone in this room to write down, it would have been a good exercise, it would take too long. Write down what your estimate of cover would be in this plot. What would you get? And I guarantee you it would be mind-boggling to everyone how wide the range of estimates would be. It would be surprisingly so. And there's a lot of data that shows that. There's been heaps and heaps of research saying how bad we estimate things. And so if I estimate 10% cover and someone estimates 30% cover, then we've got a 20% difference straight away there. I might have done the first survey, someone does a second one. We've got an increase in 20% cover between yesterday and today. It's great. But it's not real, and that's the problem with visual estimates. And so they're good, they're quick, they're simple, but how can you rely on the data? And that's the challenge. It's not saying that visual estimates are always bad, but you've got to remember that they're pretty dodgy in most cases. So the alternative is using a category. So many people here would be familiar with Blanc cover score. It's breaking up the 0 to 100% into categories. R and plus up the bottom for a couple of individuals, and then 1 to 5 for having more cover. And there's lots of different, everyone tweaks it. There's a modified broad and Blanc that they use, and so there's lots of different slight variations on the same thing. And so for a change in time at 2 sites, we've gone and given it a category 2 in the first survey and a category 2 in the second survey. So there's no change at this site in cover. But in reality, we actually can see that there's the bounds for category 2 is 10 to 25% cover. So it could be anywhere in that range, assuming we got the category right, which is possible that we got the category wrong too, but let's assume that we got it right. And so it's possible that red line there, there actually is no change in these sites. It's about 12 or so % cover in each one. That's good. That would be a good result. But it's also possible that on the first one, it was only 10% cover, and on the second one, it was 25% cover. Still fits in category 2, but we've had a more than double increase. Likewise, it could have started from high and then got more of a half decrease, and we just can't see that. So our category 2, no change, and we've actually lost half of the cover of this thing that we care about and we're monitoring. So again, we need to be careful about this type of data collection too. Categories can be helpful and they can be simple and that sort of thing, but how are you going to use the data? Do you care if you miss this variation? Or do you only care if you're really picking up, you've gone from very low to very high or high to low? If that's all you really need to know, and if it's a little bit of change up to 20, or double or triple at the lower levels, then that doesn't really matter. You just care about the catastrophic changes or huge increases, then by all means use a simple one like that. But this has challenges if you want to know the number. So back to our quadrats, what do you do if you want to get a system that's not a visual estimate? There's a few things you can do. An example is gridding up your quadrat. This is pretty commonly used, and so there's a couple of ways of doing it. You can count the intersection points. So this is usually that's a polypipe frame or whatever it might be, and you put grids across it with string, and at every one of those intersection points is a point data collection, and you say 5 of those points touch my plant that I care about, and I've got 16 points total. 5 out of 16 points is 31% cover. And that might not reflect your estimate necessarily of this patch, and it might not be the exact answer, But if everyone in here did that count out of 16 points, they're all going to get the same number. And that's the key, that it's repeatable. And that's really important for monitoring. So whether I do it or someone else does it, we're all going to get the same or very similar answer when we go through time. And that's a really important part of monitoring. Just as a highlight there, that's different. The point intercepts is different to saying, is this thing present within one of your little squares? So it's actually, there's a bit of plant in 14 of 25 total squares. That's not cover. So you can't say that's whatever that number is that percent cover. It doesn't work when you're doing presence in an area, because in some cases it's only occupying a tiny bit of that square, and sometimes it's occupying the whole square, and so it doesn't translate. So you need to keep that in mind as well. Transects, for just a quick run over, it's a line. You collect data at points along the line. This is me looking excited about doing points on this line, and you can see why I'm so excited. So this is next to a river, and so in this case, there's doing points along this tape. The pole in my hand is my data point that helps me see at this point anything that touches this pole is what I'm recording. And so in this case, I'm mostly recording the ground layer. But it's good because I can say I can estimate the amount of the gravel or the leaves and that sort of thing. And maybe I've hit a plant at one point along that line. And the good thing is that if I do it or someone else does it, we get a really similar answer. And again, it's the repeatable part of this process. It's not, if I just guessed the amount of leaf cover through that site, it's going to be wildly different to maybe. It could, by absolute fluke, be the same, but it's more likely to be quite different. So the downside of transects and small quadrats is that they're not very good at recording small or rare things. There's big gaps between transects. You might just doing your points along a transect, you just jump over something. It's like, it's just there and I can see it, but I'm not touching it. And so if there's something that's very low cover or it's you don't see it very often, it might miss it. And so if you have a real interest in the things that are really rare in your site or really small, then maybe transects or pointing type approach is not the best thing. You might think about something else. And it's slow. Collecting that data it's good data. It's good quality data, but it's slow and it's expensive. So can you afford that with the resources that you have? The upsides is that it is repeatable, less objective, but you can also record gradients. We keep the data as you go along the transect. We've gone from a wetter place to a drier place. We can see that as we go along that transition, things are changing. There's other things you can do with data of that type. Same thing with quadrats going along. So just a reminder, so think about the data. What is the data what are you going to get? What is your harvest that you're going to get from the thing that you've planted? And what are you going to do with it? It is really important. You don't want to get to the end and, as some others have mentioned, and not have what you need. So here's a little case study just before we finish. Here's this spot on a river. I do lots of monitoring of rivers, so there's a bit of anyway. So there's a patch here that we care about this with some plants on it. We want to monitor these types of patches along the river, and there's a bunch of ways we can do it. So how do we go about it? We could lay transects across this patch and collect data on points and that sort of thing. That could work. If we care about rare things, then that might not be appropriate. But it does have the benefit. We can look at the gradients from the water, from closer to the water to further away from the water. Maybe there's something going on there. This is an option. An alternative could be quadrats within this space, small ones. Where we can go and we can randomly locate them through the site, or we can put them in lines again and get to that question. So what's our question? Do we really care about that transition? Do we care about what? And so it's going back to your objectives. What are your targets? What are you trying to achieve? What do you want to do with the data once you've got it? An alternative is maybe actually lots of little samples is really hard to record, and with monitoring, if I go to a quadrat and then come back again and I go to the quadrat, but I don't really know where it was, and so I go nearby. There's actually looking at something very different, and that's a real problem for my site. So it's really hard to relocate my quadrats if I do lots of small ones. So I can't do that. I can't put pegs throughout the site and find them all again. So I'm just going to do one big one. I know that I can find that one again. I'm going to put my effort into that. I don't really care that I'm missing bits around it. The priority is to get a site that I can go back to and collect data within that. That's good, but it's a bit of a bigger space. It's hard to, I've got to do some collection in that. So maybe I run transects through that area to collect systematic data within there. Any of these things are options, and none of them is wrong. Again, it depends on the purposes of what you're trying to achieve. So once you've done that, you need to refine. You can't monitor everything. You can't just go collect everything. Think about what the data you need. So actually in that spot, I only really care about one species. I don't have to collect data on everything. Maybe I don't care about what's on the ground. Maybe I just care about how many individuals of one specific species there are in that patch. I just go collect that. It's a lot quicker, it's easier. I don't need to do everything. Maybe the response that I'm caring about only occurs over a couple of years, and after that, I don't care anymore. So I don't need to set up a 10-year monitoring program. I need good information over the first couple of years. Also, don't leave data gaps. The last thing you want to, so this is the one is, don't do everything necessarily. You can collect more data than you need. The other one is don't forget data as well. And so there's a real competing thing. You're out there, you're collecting data, you can get this information. You don't want to come back and be through your analysis and it's like, oh, geez, we don't have this information about, we're trying to do this grazing and we don't know about what type of grazer it was on that property next to it. I've got to now go back to all my sites and collect extra information, and it's a very costly process to do. So make sure you're collecting everything that you need to do that. Some examples are around water. Rainfall is usually pretty easy. You can just get it from BOM or something like that. But maybe you're looking at a wetland and I was like, yep, that got wet. Going back, looking at the data, and it's like, actually, I really think there's a different response depending on how deep that wetland got. But I don't have the data on that, and now I can't ask that question or answer that question. So if I had have collected some depth data or something else, then that would have been a good win. Soil, revegetation is a really common one. Like the last talk was quite good about it's better ways of putting in that output data. So how many plants did we plant? Were they mostly shrubs or trees, and how densely do we plant them? And so that sort of information is really valuable to coming back. This one was more successful, but or there's more canopy cover there than there is this one, but we planted twice as many trees there. So we need to know that. If we don't have that data, we can't look at that. So last page, summary. Monitoring, rarely easy to do or cheap to do. It can be pretty expensive, and that comes back to that point of on the 7 deadly sins. You don't have to always monitor everything. In some cases, you just don't have the budget to do it properly, and so you might as well not do it. That's not at selling monitoring very well, but usually you do have to, and so you do it properly and that sort of thing. So, but need to think about it. You don't want to waste all your money and get to the end and you don't have what you need. If you think about it cleverly, you can get really the right sort of data and be able to inform something. Start with your objectives and targets, the same as that whole cycle of what we're talking about with the open standards and all these other frameworks for how you go about it. It's the same applies to the monitoring piece in itself. What's your monitoring objective and your monitoring target, your indicator, your KQs, KAs. Just their thing about avoid monitoring something broad or something not so measurable like vegetation condition. So that's okay in itself. Not saying that don't ever monitor vegetation condition, but think about what you're really wanting to get at. Maybe it is everything about condition that you care about, but maybe it's only one piece of it. So if you come up with that, try to dig a bit deeper and refine your question. Select the methods that will get you the data you need. If you start something and it's really not working, change it. This is really important. Try to remove uncertainty. This is really quite crucial. It's the same as putting in a funny design that's not going to collect the right data you need. But if you collect data and you collect it time two, you've done this whole big monitoring program, and then you've just got really uncertain values and you just can't show anything. That happens really often in monitoring programs. So you get to the end and you actually, we've got data, it was designed well, but we just don't have enough to be certain about our answer, and or there was a small change and we're not really sure what it is. So if possible, remove your uncertainty through your sampling design. And think about how you'll analyse and use the data. If you care about the gradient or you care about the depth in the wetland or something, make sure that you collect that data and you think about how you're going to analyse it. If you want to compare different grazers, then you need to get replicates of grazers for rabbits and cattle and sheep and whatever it might be. If you care about that sort of question, collect that data. Collect enough data within each of those categories. Then you will be able to actually follow through your analysis all the way through to the end. Trial your method and update. No, I don't think I've ever seen someone design a monitoring program from scratch and then go out, test it first go, and it's all been perfect. That will never happen. And so there's always something that changes, even if it's just your data sheets or something. Go out, test it, give it a whirl. Fix up the things that are a little bit out. If you collect your data and it's too uncertain, all right, we're not going to get what we need here. We need to collect more data or change our approach. And finally, a bit of a bugbear as well is to collect and store the data properly. Don't just chuck it in a silo and then just leave it and then not do anything with it. Store it efficiently. If it's an Excel file, make sure your column names are sensible. Make sure that file is put somewhere that's not going to get lost or deleted by someone. Put it in the cloud or whatever it happens to be, and think about how you're going to use it. If you don't have the capacity yourself, then talk to someone who does. Data is a really valuable resource. You're producing this thing, you've got this really valuable thing. Don't just, this Excel spreadsheet is amazing. It could be thousands and thousands of dollars worth of numbers in an Excel spreadsheet. So treat it with care and you'll get a lot out of it, I suppose. That's it.